Welcome to this explainer. Today, we're tackling something that usually causes a ton of headaches for med students and professionals alike, and we're totally transforming it. We're going to take those chaotic, microscopic blobs of histology and flip them into a crystal clear, highly organized architectural blueprint.
Seriously, why does histology always feel like an endless list of isolated details? If you've ever stared down the barrel of a microscope and just felt completely overwhelmed by a sea of pink and purple shapes, your frustration is incredibly valid. It's so easy to get lost when you're just trying to memorize isolated parts without seeing the big picture. Okay, let's dive into this. To make sense of it all, we really just need a master key. And that key, it's understanding the universal dual arrangement that governs practically every solid organ in the human body. Here is our roadmap for today. 1. The solid organ blueprint. 2. Stroma, the supportive framework. 3. Parenchyma, the functional tissue. 4. Cellular arrangements and staining. 5. The rule-excluding structures. And 6. The outside-in method. Let's get right to it.
Starting with Section 1, the solid organ blueprint, also known as the master key.
So this is the overarching structural dichotomy that defines literally every parenchymal solid organ you'll ever look at. I want you to think of a solid organ exactly like a massive office building. On one side, we have the stroma. This is your supportive connective tissue framework. It's the rigid steel scaffolding, the concrete walls, the long hallways, and the plumbing that keeps the whole building standing and continuously supplied. Then on the flip side, we have the parenchyma. These are the specialized workers inside the building doing the actual functional jobs, stuff like filtering, secreting, processing. Every single solid organ relies on these two distinct sides interacting perfectly together. Moving on to Section 2, stroma, the supportive framework. We're starting at the very outer walls of our biological building. Now, at the outermost level, the stroma usually forms a tough connective tissue capsule, but biological architecture is wonderfully diverse, right? Let's look at a few fascinating exceptions, because this is where form perfectly matches function. For example, the thyroid gets a double capsule for extra structural routing. The palatine tonsils, they have an incomplete capsule. They're protected deeply, sure, but left totally exposed on the surface so they can intentionally pick fights with foreign antigens. Then you have the islets of Langerhans, which are completely unencapsulated, just floating directly within the pancreas. Meanwhile, the testes requires a rigid, thick, collagenous shell called the tunica albuginea. Bone marrow trades a fibrous capsule entirely for a rigid, bony container. And finally, the central nervous system relies on its own highly specialized meningeal casing. As we move inward from that outer capsule, the stroma reaches deep into the organ to create internal partitions, and we call these septa or trabeculae. These structures brilliantly compartmentalize the continuous mass of cells into distinct lobes and lobules without crushing the functional tissue. They basically act as safe, deep conduit pathways for nerves and blood vessels to travel. A really fantastic example here is the spleen, which actually has fibromuscular trabecula. That means these walls literally contain smooth muscle that can physically contract to squeeze stored blood right out into circulation. How cool is that? Now, deep down inside those lobules, surrounding the individual cells themselves, we find delicate type 3 collagen forming a dense reticular network. You'll see this heavily in the adrenal glands, spleen, and liver, where rapid exchange is absolutely vital. Now, what's really interesting about this slide is the thymic exception. The thymus essentially takes the collagen playbook and just throws it out the window. Instead of collagen fibers, it uses a totally unique 3D meshwork of star-shaped cells connected together. So it's a cellular framework, not a fibrous one, designed perfectly to nurture those developing T lymphocytes. All right, Section 3, parenchyma, the functional tissue. So we move past the scaffolding, past the hallways, and we finally get to meet the specialized workers of the organ. You know, the word parenchyma actually has a fantastic historical origin. It was coined by the ancient Greek physician Erasistratus, and it literally translates to something poured in beside. I just love this imagery. It so vividly illustrates how these metabolically active, functional cells conceptually just fill the empty spaces of our stromal scaffolding to give the organ its ultimate purpose. So how exactly are these workers spatially organized? Well, the most common layout is the corticomedullary architecture. The absolutely crucial point here is understanding the spatial difference between these two zones. The cortex is the outer peripheral domain. It's usually where the heavy lifting, like functional filtration, happens. The medulla, on the other hand, is the deep central core, generally serving as the conducting or neuroendocrine center. Just think of the kidney. That outer cortex does the aggressive, complex blood filtration, while the inner medulla basically collects and conducts the resulting fluid safely down right through its core.
Of course, in biology, not all organs follow the simple cortex and medulla rule. Some layouts are incredibly complex to support specialized physiology. The liver, for example, organizes its workers into these massive, repeating hexagonal lobules. Moving on to the spleen, it intertwines immune-monitoring white pulp with blood-filtering red pulp. The testis is distinct because it runs dual parallel spaces, tubules for exocrine sperm production and interstitial spaces for endocrine testosterone synthesis. And finally, the pituitary gland simply houses two entirely different developmental domains, glandular and neural, right side by side.
Let's move to section 4, cellular arrangements. This is where form meets color.
Let's see how this all builds into the physical shapes these workers form and the colors they wear under the microscope. When you're looking at these cells, you'll often see them arranged in rounded clusters. But there is a really commonly confused difference between an acinus and a follicle. Exocrine acini are grape-like clusters. The cells form a wedge shape around a tiny central hole, and they're specifically designed to squeeze their secretions right into an excretory duct. Endocrine follicles, on the other hand, are sac-like closed spheres. They don't have an outlet duct at all. Their cavity is built solely for the bulk storage of materials, like how the thyroid stores colloid. To put it simply, acini excrete, follicles store.
And this brings us to the secret color code of histology, H&D staining. Basically, the color a cell takes on tells you exactly what that cell does for a living. If a cell is eosinophilic, meaning it stains bright pink or red, it is packed with mitochondria, meaning it has super high energy and ATP demands. If it's basophilic, staining blue or purple, it is loaded with ribosomes and rough ER, making it a massive protein factory. And if it's pale or kind of washed out looking, it's essentially a warehouse where all the stored goods, like lipids, glycogen, or mucus, just got washed away during the slide's chemical processing. It's that simple. Color equals function.
Which brings us to section 5, rule excluding structures, the biological rebels. We have to acknowledge that in biology, there are always rebels that just outright break the standard rules. Boundary structures like the lip, the tongue, and the eyelid do not use our standard capsule and parenchyma blueprint. Instead, they rely on specialized musculomucosal cores. Take the tongue, for instance. It's a massive block of skeletal muscle running in 3 orthogonal planes for extreme mobility, and it's covered in sensory taste buds. The eyelid, that's a highly engineered layered shutter with a rigid fibroelastic plate. These structures toss out our simple office building analogy because they are explicitly tailored for dynamic mechanics, protection, and sensing right at the edges of the body. And finally, section 6, the outside-in method, our guide to practical slide viewing. Let's bring all this amazing theory together into a highly practical tool for examining your microscopic slides. If you want to never, ever get lost in a microscope again, you just have to follow this systematic 3-step outside-in process. Step 1, start on low power and check the outer border, or the capsule. Look at its thickness and integrity. Step 2, move slightly inward and inspect the subcapsular zone and those major stromal hallways we talked about, like the septa. And step 3, only after you fully understand the scaffolding should you drop down to high power to analyze the intralobular parenchyma, the actual working cells and their pink, blue, or pale staining. Here is exactly why this matters, and this is honestly perhaps the biggest takeaway of this entire explainer. Dropping straight into high power right in the middle of a lobule is a massive trap. Parenchyma cords look remarkably similar across a lot of different tissues. If you skip the supportive framework, you won't just misidentify the organ, you're going to completely miss critical peripheral pathologies, things like early capsular invasion, subcapsular hematomas, or early bridging fibrosis. You absolutely have to see the building from the outside before you interrogate the workers inside. By separating stroma from parenchyma and really respecting the incredible link between cellular form, color, and function, the microscopic world stops being a chaotic mess of shapes and becomes a beautifully readable map. So now that you possess this architectural blueprint, what hidden pathology will you uncover when you look at your very next histology slide? Happy exploring.